forum is out of town, so I want to introduce Dr. Tony Muslin. He's the Oliver Langenberg Distinguished Professor of Science in the Practice of Medicine, Professor of Cell Biology and Physiology, Director of the Center for Cardiovascular Research, and the Director of the Cardiology Research Fellowship Program. Dr. Muslin's research focuses on molecular causes of hypertrophy and congestive heart failure. His studies have helped to identify signaling molecules responsible for the development of cardiomyopathies and diabetes, and for linkages between hypertrophy associated with hypertension. In addition, he has identified a regulatory switch which differentiates the normal, healthy hypertrophy associated with athletic training from the deleterious sort associated with hypertension and diabetes. Dr. Muslin received his undergraduate degree from Yale, his medical degree from Harvard, and he did a fellowship at UCSF. From there, he came here, and now he's here with us. Thank you. Thanks a lot. Yeah.
Well, I'm sorry to hear that Dr. Moody is in Switzerland drinking Dom Perignon and eating caviar, but I guess you can see this on the videotape. Hi, Yorm. So, the title of my talk, unfortunately, is the exact same title as last week's talk, and if I had known Dr. Mann was going to use the same title, I would have changed this slightly. But I'm going to talk about pathological cardiac remodeling after myocardial infarction, and I think that differentiates my talk from Dr. Mann's talk. And I'm going to really emphasize the relationship between myocardial infarction and the eventual development of heart failure. So that's really the general area, and I'm going to tie in this clinical problem, which is cardiac remodeling after myocardial infarction, to the basic science that I work on, which is signal transduction, growth factor signal transduction. And please interrupt me if there's any questions. So like I said, I'm going to focus today on acute myocardial infarction and responses that occur in the heart after the MI. And this is just a cartoon showing, or a picture showing a heart, autopsy heart, let's say, after a myocardial infarction. You have a coronary artery on the outside. You have several coronary arteries on the outside of the heart, and they're embedded in this fat. That's every human heart has fat on the surface of it, and the arteries sort of pierce into the muscle and supply blood and oxygen to the muscle as it works hard. And if there's a clot, usually at the site of a atherosclerotic lesion, and often when in the setting of a lesion that's ruptured or cracked open, then you can get a clot with platelets, then you'll get a myocardial infarction distal to where the artery is clotted. And the usual clinical symptoms of a myocardial infarction are heart attack, or chest pain, arm pain, dyspnea, shortness of breath, nausea, diaphoresis, sweatiness, palpitations, and malaise. And when you go to the emergency room at Barnes Hospital or other hospitals in the area, you'll find an abnormal electrocardiogram, usually in an elevated troponin. And so that's just the clinical picture of an acute myocardial infarction. And then if you go to Barnes Hospital and Dr. Hurrian is in the emergency room, I saw you, he will look at the EKG and say that you need to go. Let's say you have evidence of an acute ST elevation myocardial infarction with an abnormal EKG. He will call up Dr. LaSala or somebody else in the cardiac catheterization lab, and they'll take you down to the cardiac catheterization lab, which looks like this if it's new, and they will a catheter will be placed up your femoral artery, and then in this Judkins left catheter will shoot dye down one of either your left or your right coronary arteries, and you'll see this image with X-ray camera. And so if they see an occlusion, so this is what a normal cath might look like, where this is the left anterior descending system, and this is the circumflex system, and if there is an occlusion, you might see just a little stub like that. And so let's say you've come into the hospital with chest pain, and you have evidence for EKG, for MI by EKG, and you have this cath, then the procedure that typically occurs is that a stent is placed, and so a catheter is placed in the site of where the clot is. You put a wire through the clot, and then you can blow up a balloon and deploy a stent. And they have fancy stents now that are drug-eluting, that have various drugs, especially everolimus is the new one this year, but tacrolimus and sirolimus, and there are other drugs as well that have been embedded into these Taxol, other drugs that have been embedded into the stent designed to prevent restenosis. So that's a happy ending, except for the fact that most patients, even after they get the stent, still have had a heart attack. And especially if you don't get to the cath lab very quickly, you could have a fairly sizable heart attack, despite the fact that a stent has been deployed and the artery has been opened up. And so that brings us to the issue for today, is even with best possible therapy, often heart muscle dies rather quickly, and so the 30, 60, or 90 minutes it takes to get you to the cardiac catheterization lab and find the clotted vessel and to open it up, you'll end up with a heart attack, and that there's a central area that's dead. And the problem is that if you do end up getting necrosis of an area of the heart responds in a way that seems to be physiological, or at least at first attempts to be physiological, but unfortunately changes occur that end up being non-physiological and deleterious. And so from this review in JCI, you start off with a heart attack, and it turns out that in many patients, for a variety of reasons, many of them biomechanical, the heart attack grows larger. There's extension of the infarct. The part of the heart that's not involved at all can often get hypertrophied, and then you can also often end up with a few weeks later or months later with this dilated, poorly functional heart. So you could start off with a 20 or 15% myocardial infarction and end up with a 40% one a few weeks later if you don't get appropriate therapy, and even with appropriate therapy, you could still get bigger.
Tony, can I ask a question?
Yes,
just
please.
on that one? So, necrosis versus apoptosis.
Right.
Is to say, some years back, no one talked about apoptosis in
Right.
this setting, but today one hears that.
Right.
So can you say a bit more about it?
Yes, I'm going to talk a lot about apoptosis today, and that's what I'm going to focus on. But the trouble is that, clearly in the acute phase of the myocardial infarction, what you're mainly dealing with is necrosis. But it actually, in the remodeling stage, some of it is still probably necrosis. So I would agree with you. Acutely, everything is probably necrosis. But in the subacute phase, there's a mixture of necrosis and apoptosis, and there's this new thing called programmed necrosis now. And so it's kind of a continuum. And I think the field is rapidly evolving. There are all sorts of new molecules that come out every year, such as this. There's molecules involved in the mitochondrial transition pore, and that's gone up and down in popularity, whether that's related to necrosis or apoptosis. So it's a highly dynamic field. But it's a good point that you have to consider both of those mechanisms. And so let's just say a little bit more about pathological cardiac remodeling after myocardial infarction. And this is the most important structural event after MI, and it's derived from intracellular and extracellular structural changes of the myocardium, and it elicits structural changes of the LV wall. And the intracellular structural changes include apoptosis of cardiomyocytes in the infarct border zone. So you can't really see well here on this image, but on the edge of the infarct, where there's a lot of tension, there can be apoptosis. And there's also further mechanical load on the LV, like if you have high blood pressure, that could cause worsening of the situation, it could cause dilatation. And it's thought that LV remodeling is a predictive factor for mortality and morbidity of the patient with MI. So there are many biological reactions that are involved in the time course of remodeling after an ischemic episode. I mentioned this issue of the local ischemia and myocardial cell death at the time of the initial infarct, which is necrosis. And then there's oxidative stress and inflammatory reactions in the injured myocardium, and that can lead to apoptosis and hypertrophy. There's a variety of cardiodepressive reactions due to the production of reactive oxygen species and inflammatory cytokines, and this is something that Dr. Mann probably talked about. There are changes in extracellular matrix following activation of matrix metalloproteinases, and there are structural changes in the myocardium in response to mechanical stress. And finally, synthesis of collagens and fibrosis. Looks a lot better on this screen than it does up there in this part. Why? Okay. And so what about the role of reactive oxygen species? In the ischemic myocardium, reactive oxygen species are generated, especially after reperfusion. And the major sources of reactive oxygen species in the ischemic reperfused myocardium are mitochondria, xanthine oxidase, and phagocyte NADPH oxidase. And ROS, or reactive oxygen species, directly injure the tissues, and they're key molecules for inducing cell death, and I'll talk more about how that works. And detrimental effects of ROS are clearly demonstrated by the fact that transgenic mice, which overexpress superoxide dismutase, you can reduce infarct size. So there's something about reactive oxygen species in the infarct and peri-infarct time. And so what's the current therapy to prevent remodeling? And so current post-infarction therapy to limit cardiac remodeling includes ACE inhibitors, or ARBs, angiotensin receptor blockers to reduce vascular afterload, limit LV wall stress, and reduce intracardiac angiotensin II signaling, beta blockers to reduce heart rate and myocardial oxygen demand, aldosterone antagonists like eplerenone to reduce LV fibrosis. And the issue is, can we do better in other druggable targets? And so, in order to address this issue about remodeling after myocardial infarction, several years ago, the mouse cardiovascular phenotyping core in the Center for Cardiovascular Research over in the medical school, we established a myocardial infarction model in mice where we suture the left anterior descending artery in a mouse and get about a 30% anterior myocardial infarction. And Carla Weinheimer worked on this, and also G. Ren worked on this in my lab, and Carla is part of the core. And so what we wanted to do is to be able to set up a situation where you have non-lethal, reproducible infarcts, and then we can follow the mice in certain genetic settings or with drugs and see if we can modify the remodeling after the initial MI. And I just want to point out, this is a different model than a lot of times if you go to meetings, what you'll hear about is this ischemia or perfusion. You cut out the heart of a rat, or a guinea pig, or a rabbit, or a mouse, and you just stick it on a Langenberg machine, and you make it ischemic for a while, and then you give it oxygen back, and you look at, and then you stain it. And so that's a completely different ex vivo model. And this model, in my opinion, is more representative of what the real problem is, which is humans who haven't had their heart cut out and stuck in a Langenberg machine, but who've actually had a myocardial infarction, and then they get remodeling with time. And so that's, but I have to say that there is a debate in the field, and we can certainly talk about this later, about which is the more appropriate model of remodeling after myocardial infarction, that acute ex vivo ischemia or perfusion, or this in vivo situation. So, but this is the approach that we have used. And the sort of the most famous person who is in favor of the ischemia reperfusion in the ex vivo approach is Roberto Bolli at Louisville. All right. So anyway, so this is the way we've done this to look at this remodeling situation. I talked about all this stuff. And one thing that I should say that's the stated major advantage of the ex vivo approach, where you hang a heart and then make it ischemic or non-ischemic, is that you can really control the situation. You know how long the ischemia is, you know how long the reperfusion is, you can stain the heart, you know what the area at risk is, and so forth. But what we've done is, with Attila Kovac's help, is really try to quantify the exact size of the initial infarct. And either a couple of hours after the suture is placed around the LAD or the next day, Attila can look carefully by two-dimensional quantitative echocardiography, and there's a variety of indices that he can use. So-called segmental wall motion stroke index, where you look at a variety of different segments of the LV and say how much that different segment is moving. And I mean, it's not a perfect situation, but it's a pretty reliable quantitative estimation of how big the MI is. And obviously this is a critical factor, is can you normalize these MIs initially? Because if you have different sized MIs, that would affect all of your data further downstream. So anyway, I'm pretty confident that Attila has been able to reliably quantify the initial infarct size, and then you wait 7 or 14 days later and see how much things have changed. And so just to go back to the signaling and what we're looking at, I mentioned that in ischemic tissue you can generate a lot of reactive oxygen species. And why is this, how does this relate to signaling? And it turns out that there's this very interesting protein in a lot of cells, including heart cells, that is a sensor for sort of the amount of reactive oxygen species in a cell. And that one of those proteins that senses that is thioredoxin, which we've heard about before. And thioredoxin has these SH groups, and in the absence of reactive oxygen species, it turns out that thioredoxin can bind to various kinases and inhibit them, such as this one called ASK1. or apoptosis signaling kinase. You can see where I'm going. This is inhibited by TRX, by thioredoxin, in the absence of reactive oxygen species. But if you generate a lot of reactive oxygen species like you would with ischemia, then you get these disulfide bonds, and the thioredoxin falls off the ASK1, and it can dimerize and become active. And then once these thioredoxins have fallen off, you activate the ASK1, and then you get either these two pathways, the junk MAP kinase or the P38 MAP kinase, and they do a variety of things in the cell that I'll talk about later. And so this is just the paper that sort of established this fact from this Japanese group a few years ago in EMBO journal, where they found that mammalian thioredoxin is a direct inhibitor of apoptosis signal regulating kinase 1. And it really linked, it really showed, I think this is a beautiful molecular example of how something like reactive oxygen species could be directly linked to signal transduction cascades. And one other thing to point out is that when you do have reactive oxygen species and you activate the ASK1 and then that gets P38, that P38 is important for cell death in this way. And this is an important paper also in EMBO journal a few years ago, where it was found that actually P38 MAP kinase, one of its major targets is the P53 protein, which is very important in promoting apoptosis in response to a variety of stressful stimuli. And so that's what we think one of the reactive oxygen species, a major pathway in heart cells, is reactive oxygen species activate ASK1, P38 to P53. And so then P53 is, as you've probably heard about before, is very important for inducing cell death in a variety of circumstances. And it's a transcription factor that leads to the production of these very potent pro-apoptotic proteins like BAX, PUMA, and NOXA. And this is just a cartoon looking at all the different things that regulate cell death through apoptosis. Too complicated to discuss in detail, but just the point is that these pro-apoptotic proteins, BAX and PUMA, can go to the mitochondria and lead through cytochrome C to the activation of caspases, and then chromosomal degradation. So anyway, to return to our studies, I've talked about this P38 pathway, and the fact is if what I'm saying is true, then hypoxia should cause activation of P38. And a few years ago, G. Ren looked at mice where he gave them a myocardial infarction and just cut out the heart and looked at activation of P38. And he found that in the border zone, on either side of the infarct, that there was dramatic activation of the P38 MAP kinase in the heart tissue itself. And so there is, and there are other people who've done this as well, and there is both acute and sort of more sustained activation of P38 MAP kinase in the border zone of the left ventricle after a myocardial infarction. So this enzyme is activated. And so to study the role of P38 in the remodeling process, we used a transgenic mouse model system where we used one where there's an inhibitor of P38, a dominant negative form. And so the way we make these transgenic mice is we take a promoter, a cDNA encoding a promoter, so we have the alpha myosin heavy chain promoter from Jeffrey Robbins at the University of Cincinnati, and we link that to a cDNA encoding dominant negative, either P38 alpha or beta, and I'm just going to talk about alpha today, which is the dominant one in heart. And transgenic mice were generated with high-level expression. The construct was generated by Yibin Wang, who's now at UCLA, and these mice appear normal. And just to show you that when we have these transgenic mice and you cut out the heart and you do a Western blot looking for the levels of the P38 protein, there's a lot of this dominant negative protein in the hearts of these transgenic mice. And not surprisingly, if you overexpress this inhibitor, when you, if you don't have a transgenic mouse, just a wild-type mouse, and when you do something that does activate P38, like pressure overload by banding the aorta, so sticking a clamp on the aorta, you'll get increased P38 activity, and that was blocked with these dominant negative transgenic mice. So we have a transgenic mouse model system, and it's expressed in the heart, and it blocks P38 activation. We did some initial studies with this P38, and we were kind of disappointed. At the time, we thought that P38 might be important for cardiac hypertrophy. We spent years and hundreds of thousands of dollars trying to show that P38 was critical for cardiac hypertrophy in response to hypertension. It was a big zero. It's not important. So P38 has nothing to do, apparently, with cardiac hypertrophy. So when you really inhibit it with either of these, we really didn't get we were expecting less cardiac hypertrophy. We certainly did not get less cardiac hypertrophy. So this molecule is not that important, or is not important at all, for the hypertrophic response to pressure overload. So then we wanted to see whether it had something to do with this remodeling that I've been talking about. And so we did this was the first paper where we looked at remodeling, and you know, we did the myocardial infarction, and a couple of hours later, Attila did the echocardiogram, and he found identical initial infarct sizes, about 30%, 31 here, 29.3 there. So these were statistically similar, and so the initial infarct sizes in the non-transgenic and the mouse that had the dominant negative P38, the initial infarct sizes were the same. And then we waited a week, and then a week later, lo and behold, the mouse that was transgenic with less P38 activity, the dominant negative one, had a much smaller infarct area a week later, so 45 versus about 30. So it's really quite a dramatic difference. And you can see here in the wild type mouse, here's infarct. This is trichrome stain. This whole area is infarcted. And this is not exactly the same level, but in the LV, but there was a much smaller infarct.
Is it true, it just seems from this example that hypertrophy is much larger in the
It was not, this is just a different, slightly different, the trouble is getting the exact right angulation, and I think this is just at a different level of the LV. So, but if you just looked at apoptosis, what we found is there was also much less apoptosis in the border zone area. So this is looking at cleaved caspase 3, and I don't think there's a difference in hypertrophy because we weighed the hearts and they weighed them. I don't think there was a difference in weight. There's less caspase activation, and this is a tunnel, so this is positive. These fluorescent are just positive nuclei for tunnel, and there was less tunnel positivity in the dominant negative P38. So our conclusion from that study was that P38 was important for this remodeling process. If you inhibited it, you reduced the pathological remodeling. And were we right? Was that finding 4 years ago correct? Well, I'm happy to say that in a Nature Materials paper just a few months ago, another group used a P38 inhibitor. It was kind of an interesting paper. That's why I'm bringing it up here. And they used these polyketal microparticles. This is what they look like under transmission. Or under EM of some kind of microscopy, and they stuck in this SB compound, SB239063, on these polyketal microparticles, and then they just injected them into the LV at the time of the myocardial infarction. This is in rat now, and so this is a P38 inhibitor drug that they use these microparticles, and what they found is similar to what we found. Is that if you look at fibrotic area, this is MI plus polyketal P38, the area of scar is much less. I think it's 7 days after the myocardial infarction. And so, and I don't know which one is, but whichever one looks the best is the one that had the polyketal particles. But in any case, this is this is the conclusion of their paper, that it worked, and that they reduced pathological remodeling. So it seems like maybe a P38 inhibitor drug would be a useful therapy for people at the time of myocardial or after myocardial infarction. And so what I've told you so far is that in terms of pathological remodeling, there's reactive oxygen species generation in the infarct and infarct border zone. That inhibits thioredoxin, which then falls off of ASK1, which is activated. You get activation of this P38, P53 pathway that results in cell death and pathological remodeling. And I've been interested in this other family of proteins, and it turns out that these are two of the major regulators of ASK1. So this other family is called 1433. And so the two big inhibitors of ASK1 in a cell are 1433 proteins and thioredoxin. And they bind to different areas. Now, I actually don't know what gets 1433 to fall off of ASK1. So that's still a little bit mysterious. But these are both binding to ASK1 and both inhibit. And so we also did some work with 1433 related to this issue. And I'll just tell you a little bit about what 1433 proteins are. it's a family of proteins. Every cell in the body has 14q3 in it. Every animal cell in the world has 14q3 proteins in them. Yeast, plants, and not in bacteria. So after bacteria, once you have eukaryotic cells, you have 14q3. And there are 7 isoforms in mammalian organisms, and they're encoded by separate genes that have Greek names. And these proteins are about 30 kilodalton. They're acidic, and they form homo and heterodimers, and they regulate signal transduction, apoptotic, and checkpoint control pathways. And I've been interested in them for about 15 years. And so when I got to WashU a few years ago, I had figured out, before I came here at UCSF, that 14q3 proteins bind to this RAF protein kinase. And when I came here, I worked with Andre Shaw, and we figured out that actually the way 14q3 works is it binds to phosphoserine motifs, especially this motif. And then Lou Cantley's lab figured out a second motif that 14q3 proteins bind to. And this is what a 14q3 protein looks like, a dimer crystallized, and it binds to these phosphoserine motifs in this little corner here. So it can bind to 2 motifs at the same time. And so that's what they look like and how they work. And it turns out that in signal transduction, 14q3 regulates the MAP kinase cascade and also regulates this ASK1. And Haiyan Fu had this paper where he showed that 14q3 binds to ASK1 and inhibits its kinase activity. So to study the role of 14q3 in remodeling after MI, we generated knockout mice. Fortunately for us, Genomics, which I don't think exists anymore. It was this big grant to make knockouts for everything. But they had made an ES cell line that had targeted disruption of the 14q3 tau gene. So it turns out that Jeff Lau, who's an MD PhD student in my lab, was making the construct, and he just happened to be looking on the Genomics website, and he said, well, there's already a cell line that's targeted, Tony. Why do I have to work on this for another year? So we'll just call them up, and it's just a few $1,000s. So we called them up, and actually they sent us the cell line, and then we had it injected into blastocysts and generated chimeras. So Jeff got out of a year of work, and he made the knockout mice. But it turns out that the knockout mice don't live. They die around embryonic day 12. They have a variety of growth defects. But the heterozygous mice were viable and fertile. So we did a variety of studies in the heterozygous mice. And so this is just to show, if you look at the heart of a heterozygous 14q3 tau mouse, and why do we do 14q3 tau and not beta or zeta? We had found out in earlier studies in other animals that tau was very important for cell death. That if you knock down 14q3 tau, for example, in frog embryos, that there was a lot more apoptosis. And so we thought this might be an important one for that phenomenon. So that's why we targeted this family member. Anyway, so there's less 14q3 tau protein in the head. And then we looked at signal transduction. I mentioned to you that 14q3 proteins in general inhibit ASK1, so if you knock it down, there should be more ASK1 activity. With more ASK1 activity, there should also be more P38 activity. And so lo and behold, wild type and Het for 14q3 tau, there was more basal P38 activation in these heterozygous mice. So this is the Het and this is the wild type, a lot more phospho-P38. There was also a lot more ASK1 activity. Now, it's kind of an inverted situation with ASK1. When it's phosphorylated, it's inhibited. And when it's dephosphorylated, it's active. So that's a little unusual. But it's the opposite direction, but it's the same ultimate answer, that this is active now with less phosphorylation, and this is active with more phosphorylation. So what's the story? If you knock down 14q3 tau, does that sensitize heart cells to cell death by apoptosis? And so one way we did this is take cultured adult cardiomyocytes and give them hydrogen peroxide, generate reactive oxygen species, and we found this dramatic increase in apoptosis measured by the kit. I don't have that on the side here, but a kit that looks at nucleus DNA that's been clipped. And so these heart cells are dramatically sensitized to apoptosis. So then we did that myocardial infarction experiment, and same exact initial size of an infarct a few hours after the MI, and then if you wait a week, we got this result, which is that in the wild type mice, this was the infarct size a week later, And in the knockdown or heterozygous mouse, the infarct was 55% versus 39%, so dramatically increased. And you can see how diseased this looks. And it looks a lot better on my screen, so I'm not sure why that is, but I've gone from Dell to Apple, so maybe that's the reason. And just to confirm that there is increased apoptosis with this 14-3 knockdown, we looked at cleaved caspase 3 in the heterozygous mouse in the infarct border zone, and you can see increased activation of caspase 3 compared to the wild type in the infarct border zone. And similarly by tunnel, where we saw more tunnel-positive nuclei. You can tell there's more because there's more red arrows. So you don't have to look at the data, you can just look at the arrows and make sure it's right. If I put enough arrows there, you can't see the underlying things. But anyway, we thought that there was more apoptosis in the heart muscle in the border zone in these heterozygous mice. And so this is the conclusion from this pathway, and that is that we have two inhibitors of ASK1, thioreduction and 14-3 tau. When active, ASK1 activates this P38, P53 pathway that promotes cardiomyocyte death and pathological remodeling. So agents that inhibit this pathway, either by inhibiting P38, inhibiting ASK1, stimulating 14-3, would all potentially be salutary and promote a better heart health. So I'm going to change gears a little bit and talk about a different pathway, a survival pathway. So I talked about this reactive oxygen species, ASK1, P38 death pathway. What about a survival pathway? And so this is kind of one of the mother of all survival pathways, the AKT pathway, which is very famous for promoting survival of a variety of cells, including cancer cells. And so it's this pathway is a big target in cancer biology, where you're trying to inhibit this to kill the cancer. But unfortunately, it turns out that this pathway is really critical in a lot of other tissues to promote health, like heart and brain. And so I just want to emphasize that this AKT pathway is very important for maintaining survival of heart cells. And so how does this pathway work? What it is generally stimulated by growth factors like insulin-like growth factor, or insulin, or EGF, or fibroblast growth factor, or PDGF. And these ligands, when released into the extracellular space, bind to receptor tyrosine kinases that then activate this very important enzyme called phosphatidylinositol 3 kinase, which generates a lipid phosphate called PIP3, or phosphatidylinositol 3,4,5 phosphate. And PIP3 binds to AKT with another kinase that I didn't include here called PDK to activate the AKT family members. AKT goes to the cell membrane, to the nucleus, to the mitochondria, all over the cell, and phosphorylates a variety of targets and affects cell survival. And here are some of the important targets for cell survival. BAD, BAX, GSK3, and FOXO transcription factors are all phosphorylated and all inhibited by AKT. And since these are players in pro-apoptotic pathways, pro-cell death pathways, when these are inhibited, you get survival. So what is the role of this cell survival pathway in pathological remodeling? And so the argument would be a priori that maybe this is turned off in the border zone, and that's why you get remodeling. And so we'll address that. And so I mentioned that AKT family members play a key role in regulating cell survival. We've been looking at knockout mice, and our theory for a while now has been that AKT2, one of the family members, is the most important in terms of regulating cell survival, not so much AKT1. And so this is sort of the classic paper that shows why AKT is probably important in regulating cell survival. And this is from Michael Greenberg at Harvard, where in the Cell paper a few years ago, he showed that AKT phosphorylates BAD, and that blocks the activity of BAD, which is bad because it promotes cell death. And so this is what AKT proteins look like. There's 3 family members in the body, human body, 1, 2, and 3. They look about the same. Really only 1 and 2 are expressed in heart. And the knockouts have interesting phenotypes. The AKT1 knockout is viable, but it's a little bit smaller. And what we found with Brian F.A.H.A a few years ago was that when you take away AKT1, heart muscle doesn't grow in response to exercise. But there's no diabetes in this mouse. It has normal sugar metabolism. So AKT1 really doesn't seem to have that much to do with insulin for some reason. But when you knock out AKT2, the mice are not smaller. They respond normally to exercise, but they get eventually type 2 diabetes as they get older. So it's kind of an interesting model of human type 2 diabetes. So we wanted to investigate the role of AKT2 in remodeling. We started off by looking in cell culture. And so we have our in vitro model of remodeling, which we take cultured adult murine cardiac myocytes, treated with hydrogen peroxide or UV irradiation, and we measure apoptosis. And what you can see here is that just in control cells treated with hydrogen peroxide, there's a lot of apoptosis. Excuse me, in the AKT2 knockout, there's a lot of apoptosis, which is more than the wild type cells. I'm not sure what this extra R is, but in any case, anyway, but there's a lot more. We had a lot more cell death in these AKT2 knockout heart cells. And then we did that MI experiment I keep on talking about, where we gave them a myocardial infarction, both the knockout and the wild type mice. And the initial infarct size was the same in these two genotypes. And we waited 7 days, and we looked at the infarct size 7 days later, and we got a bigger infarct when you take away AKT2. It was 0.25 here versus 0.44. Anyway, there was a big increase in the size of the infarct a week later. And Ryan measured tunnel-positive cells in the border zones on either side of the infarct, and he showed the data in this tabular form and found that there was a lot more apoptosis by tunnel positivity in the AKT2 knockout hearts after myocardial infarction. So tunnel analysis of infarct border zone 7 days after experimental MI by ligation of the left coronary artery, and there was more apoptosis. So this kind of experiment with the AKT2 knockout suggests that maybe this pathway is important for remodeling, but does this have anything to do with reality? Is this pathway in some way perturbed in reality? And to address that issue, I want to bring up another protein that we started to study recently, and this protein is called TRP3. And this seems to link what happens in MI to this pathway, and I'll tell you why. So TRP3 is a cytosolic protein that is a pseudokinase that binds to AKT and inhibits its enzymatic activity. Interestingly, there's a common polymorphism in the TRP3 gene that results in an amino acid substitution, Q84 to R, and when you have this polymorphism, you actually get increased ability to inhibit AKT. And this is common in Italy, where they do a lot of studies on this polymorphism. And when you have this polymorphism with increased TRP3 activity in humans, they have a lot more insulin resistance and cardiovascular disease. And so that's kind of an interesting human angle to this TRP3 story. And TRP3 is an inducible gene in response to ER stress and also in response to reactive oxygen species. And so what about ER stress? Does ER stress have anything to do with MI or pathological remodeling? So I've mentioned that we have this TRP3 protein. It's an inhibitor of AKT. It's induced by ER stress. What is ER stress? Does that have anything to do with MI? So just a little background about ER stress. So the endoplasmic reticulum controls, as you know, protein maturation, protein folding, and conditions that interfere with the function of the endoplasmic reticulum in cells are collectively called ER stress. And it's often caused by accumulation of unfolded protein aggregates. Hypoxia also, and reactive oxygen species also cause ER stress. And so this fact makes it seem like maybe ER stress is an important thing that occurs in pathological remodeling after myocardial infarction, because there's hypoxia and there's reactive oxygen species. So anyway, when you get ER stress, you're trying to decrease the rate of protein translation to prevent further accumulation of unfolded proteins. And you also want the ER-specific protein degrading apparatus becomes activated, and you want to eliminate denatured proteins. If this doesn't work, the cell dies by apoptosis. And so that's sort of the ER stress response or unfolded protein response. And this is a cartoon from EMBO reports a few months ago, and it shows sort of the complicated pathways. This is an ER over here. You can't see it very clearly. In any case, you get activation of various proteins like ATF6 and PERK that lead to the expression of a variety of genes that are involved in this ER response, and one of the genes that gets transcribed in response to ER stress is TRP3. So we've been looking at this ER stress response in heart cells, and we've been finding that TRP3 is in fact induced in response to ER stress in cultured heart cells in a variety of settings. This is just an example of us taking cultured heart cells, H9C2 cardiomyocytes, and giving them things that cause ER stress like tunicamycin or F.A.H.A. And when we do that, the TRP3 mRNA levels go up dramatically, 20-fold or 40-fold. So compounds that cause ER stress can cause TRP3 to be expressed in heart cells. So we use F.A.H.A, which causes the release of ER calcium stores, or tunicamycin that blocks glycosylation. Anyway, they both cause induction of TRP3. And I'm going to link that to MI in a second here. Another one that Gene Schaefer's worked on is that palmitate, which we think leads to increased reactive oxygen species generation in heart cells, also causes increased expression of TRP3. If you overexpress TRP3 in heart cells, you block signaling by that are via AKT. So if we overexpress TRP3 by using an adenovirus and add insulin and look at glucose uptake, we can have reduced glucose uptake a little bit by overexpressing TRP3. Anyway, so what does TRP3 and ER stress have to do with myocardial infarction and remodeling? So this is the linkage. So what we took, we gave wild-type mice myocardial infarctions, then we cut the heart into pieces at various time frames, and we looked at 4 hours and 24 hours after myocardial infarction, and what we found is that there was this gradual increase up to 5 or up to threefold 24 hours after myocardial infarction. So there is a relationship between MI, ER stress, and TRP3 induction. Furthermore, we looked at these other markers of ER stress, CHOP10 and GRIP78, and they all went up in that time frame, 24 hours after MI, and this is looking in the infarct border zone. So by genetic markers, there is ER stress in the infarct border zone, and it's correlated with increased TRP3 expression. So if this is the case, if TRP3 is induced in response to myocardial infarction and TRP3 turns off AKT, can we somehow figure out what this TRP3 induction is doing? Is that promoting pathological remodeling? Linking it back to the earlier issues I was discussing. And so one way to examine that is to make a mouse where we overexpress TRP3 in the heart at baseline and then give it an MI. So we're giving it like a boosted dosage of TRP3. Does that make the remodeling worse? And the answer is yes, it does. And the way we address this is by generating a new transgenic mouse where we overexpress TRP3 using that MHC promoter. And we have both 1x, 2x, and 4x lines. So when we overexpress the TRP3 in the heart, and we take the hearts and we infuse either insulin into wild type or transgenic hearts, what we found is that insulin stimulation of these hearts causes AKT activation, but much less in the transgenic animal. And this is looking at GSK3 beta phosphorylated, and here it's AKT. So overexpressing TRP3 blocks insulin stimulated AKT and GSK3 beta activation. And then again, we did the MI study where we have similar infarct sizes, actually a little statistically the same, but maybe slightly smaller in the TRP3. And then if we look 7 days later, we see that there's a dramatically increased infarct size in the TRP3 overexpressor. So the wild type, the blue is the scar at different levels, and you can see that the scar is much bigger. It's not exactly the same level, but you can see it's much bigger. If you take all the different sections and you add them up, you get a much larger 25 to whatever this is, 36% infarct size in this particular series. And so, clearly by overexpressing this TRP3 protein, which inhibits AKT, we're expanding the infarct size. We're also getting more apoptosis, looking at tunnel.
Did you try it in 4x?
We have not tried it in 4x yet. 4x may get a phenotype at baseline is the problem. The thing is that the 4x, I mean, this is like the background. There's very little TRP3 you can see at baseline. So there's a little bit, but not very much. And so the 2x is already pretty high. So we were The 4X is maybe 100-fold more protein than normal, so that's why we went for the intermediate one. But we've not done the MI yet in the 4X mice. So from these studies, we know that TRP3 is induced in cardiomyocytes in the heart in response to ER stress. Ischemia and hyperlipidemia, I didn't show you the hyperlipidemia data, but we know that ischemia in any case leads to cardiac ER stress and TRP3 gene induction. ER stress leads to reduced AKT activation in a TRP3-dependent fashion. We've done a variety of in vitro studies to address this issue. And when you overexpress TRP3 in heart, you get reduced glucose oxidation rates. There's altered expression of stress response genes. I haven't shown you this data, but this is some of the other stuff we've worked on with this mouse, and we don't have any alteration in basal cardiac function, but when we have these transgenic mice with overexpression of TRP3, they are sensitized to pathological cardiac remodeling with increased cardiomyocyte apoptosis. And so then to summarize this part of the talk, normally, in a normally functioning heart, you have release of various growth factors like IGF and insulin. They activate the insulin receptor or the IGF receptor. You get activation of PI3 kinase, AKT. This suppresses the activity of these pro-apoptotic proteins, and you get survival. And so all the time in brain and heart, you have these growth factors being released. There's sort of a constant amount of IGF in the heart. You have insulin throughout the day. There are other growth factors like neuregulins and EGFs, always bathing the heart, continuously activating this pathway. So it's not like this pathway is intermittently activated. I mean, there are times, every time you eat, this pathway gets activated, and just throughout the day, there is a basal amount of AKT activity all the time. It's not something that's just here and gone. It's pretty much omnipresent. But in response to hypoxia, you have this ER stress and the induction of this TRP3 gene, which turns off this pathway and lowers the amount of AKT activity in heart cells, and then you get this, increases the likelihood that a heart cell will die. And so how does this relate to potential new therapies? I mean, I've told you a lot of stuff about signal transduction, about our model of cardiac remodeling. How can this help the treatment of patients who have myocardial infarction? So the first thing I mentioned, that the P38 inhibitor therapy may be very effective. There are many P38 inhibitors available. A lot of drug companies have generated these. There's a thought that these may be useful for arthritis, but I think they may be helpful for the heart. And there's also this issue about local targeting. I mentioned those microparticles, but there are other ways that you could target it to the heart. And so this is certainly something that needs to be considered in the peri-infarct setting. I mean, a lot of the time with cardiovascular drugs, I'm sure you're all aware, the drug companies are very reluctant to do cardiovascular trials in humans because the endpoints are difficult to achieve. It's not like cancer where everybody's going to die. And so it's always this uphill battle with the drug companies to argue with them to try these things in this setting. But I think in the peri-infarct period, it may be, if you have endpoints like ejection fraction a month later, maybe we can get these trials going. So I think this is one way of considering a therapy for remodeling. Another way is inhibiting ASK1, and there are a variety of inhibitors of this kinase that are in development. So what about the other side? I mentioned this P38 ASK pathway, but there's also this AKT pathway. And is there ways that we could stimulate this pathway while maybe inhibiting this pathway? And so actually we could do this today. So obviously growth hormone is available. It's actually off patent, although still expensive. But growth hormone could be given. We could start a trial today on growth hormone in the peri-infarct period. The reason why people are reluctant to use growth hormone in adults, especially older adults, is that it may increase the likelihood of getting cancer. But I think for the short period in the peri-infarct period, it may still be something to consider. And so I think growth hormone, IGF, EGF, other ways of activating AKT are all possible therapies. And like I said, the growth hormone or IGF could be tried in a trial today. There are also ways that we can think about inhibiting TRP3 action. And there are a variety of ways that you can consider trying to make a small molecule that would inhibit the interaction between TRP3 and AKT2. And finally, a future plan from our lab is to sort of become completely unbiased about this and try to identify new drugs that inhibit hypoxia or reactive oxygen species-induced cell death of cultured cardiomyocytes. And with Tom Ellenberger in the Department of Biochemistry and Molecular Biophysics, there's a robotic set up to do chemical biology screens in heart cells that we are developing. And so that's a future plan to use this chemical biology technology to identify new drugs that inhibit hypoxia-induced cell death. And I just want to thank the people in my lab, and this is just two pictures from different times in my lab. But a lot of the work that I talked about with P38 was done by Ji Ren and Xiaosong Zhang. And also I talked about work from Jeffrey Lau, who is here, who did the work on 43 tau heterozygous mouse. And I talked about a lot of the work with TRP3 that was done by Joan Avery. So thank you for your attention.
So as you pointed out, ASP1 is upstream of both JNK pathway and P38 pathway. So when you inhibit P38, do you see any alteration in the JNK branch?
We have not seen increase in JNK by inhibiting P38, so we have looked at that in the past. Interestingly, there is a reciprocal relationship between MAP kinases in general and the AKT pathway. And so MAP kinases in general and AKT are mutually inhibitory. And so if you lower AKT, you increase all of the MAP kinases.
You mean ERK or
ERK and JNK go up when you lower AKT, and when you lower ERK, you increase AKT. So there is a mutually inhibitory relationship between the AKT pathway and the MAP kinases, but not so much within the MAP kinases themselves. Yeah.
So has there been any trials with antioxidants and post ischemia, or like when you stent somebody to give antioxidants or anything like that?
Yeah, I mean, there's a long history of people giving antioxidants for general health, and it's been sort of disappointing. And I think a lot of the issue, I mean, I don't know the details about those trials. I don't know, Bob, do you know any of those details about the antioxidant trials? I think they've been fairly disappointing.
They don't do them.
Huh?
I think there's been some trials, not on specifically looking at the infarct period, but related to antioxidants.
It may be we're not using we're using over-the-counter antioxidants or very weak ones. We're not really using the strong ones. Maybe something like acetylcysteine, like mucormyst, acetylcysteine. I don't know if that's ever been tried in the peri-infarct period. But that's a stronger one that we use mucormyst a lot when we're doing cardiac catheterization because it lowers the incidence of renal failure in response to the dye load, and because that's a reactive oxygen species scavenger. I think it's a fairly effective one. And so, I would have to review the literature to see, but it would be interesting if it hasn't been done to consider a mucormyst trial in the peri-infarct period. That's a relatively benign drug.
So you looked at the P38 alpha and beta. What about other isoforms?
Yeah, I mean, we don't. There are other isoforms in heart, as you're aware, and I think it was like delta and gamma are in heart, and actually they're pretty high levels in heart.
And we found they actually activated in disease in humans.
Yeah, we really, the drugs don't usually work for those unusual later alphabet forms of P38, and most of the drugs just work for alpha and beta. And so actually it's kind of an unexplored area, and it would be worth pursuing. Yeah, Catherine.
It makes sense to look at the infarct border zone when one wants to really see what's happening with remodeling and things like that. But when you look at the infarct, obviously it's a living entity also with so many fibroblasts and things like that. So my question is, have you looked at the infarct itself and looked to see whether or not fibroblasts are involved in, a lot of the signaling? And then also, part two of the question is, how much of the signaling is involved in, inflammatory responses? Because when you see something activated at 24 hours, within 24 hours of the infarct, might you have inflammatory cells that are participating in that?
Okay, so there's several points you've raised. The first point is, the infarct is not 100% necrotic, and there are viable myocardium strings. If you look carefully, as you know, if you look, you'll see strings of viable cells in between dead cells, in between fibrosis, and so it is a sort of a complicated mixture. And the only advantage of the border zone is that there you have a clear demarcation where there's no scar anymore, and so it's a little cleaner area in terms of the mixture. So that's where we tend to look there. The second point about the inflammatory cells, I mean, I think that it starts to go up pretty early. Now, obviously, you could have neutrophils already by that point, and whether they're contributing, they certainly, some of the inflammatory cells obviously increase the reactive oxygen species production and load, like neutrophils, and that could be exacerbating the situation. And I actually don't know how important inflammatory cells are to remodeling. You could address that issue. You could do bone marrow transplants and then do MI with a variety of different genotypes, and that would be an interesting study. So I don't really know the answer to the net role of inflammatory cells, or even the role of fibroblasts. I think those are important questions.
Let me ask a question. Do steroids reduce infarct size? Has
Steroids
anybody
are
done it?
extremely dangerous. The studies.
No, yeah, no, you wouldn't do it in patients.
Well, the reason you don't do it in patients, it was popular for a while to do steroids, but it turns out that the rate of cardiac rupture, I don't know if you remember this, but the cardiac rupture goes up dramatically with steroids. And that's because it's thought that the scar is not as strong. And actually the 14-3
But did they die with smaller infarcts?
Yeah, I don't know. But the 14-3 tau mice, which have this increased apoptosis, a lot of them died from rupture. So that was interesting that we found that as well. But but that's the problem with steroids, is the rupture.
So it may actually, influence fibroblasts, because they're the ones that are transformed into myofibroblasts, which lay down the collagen. So if you have an imbalance between how much collagen is deposited, because the whole thing with MMP activation and that balance.
Yeah, no, I'm sure that, the fact that steroids do cause the rupture argues that inflammatory cells are probably contributing to fibroblast-generated collagen production. And that you want scar in the place where the cells are dead, or else you're going to rupture. And that's part of the process. But what you don't want is an expansion of the infarct. You want the scar to be nice and firm, but you don't want the infarct to get bigger. And you also don't want the heart to dilate. And so those are the things that you're trying to prevent. Whether or not the hypertrophy is bad, I don't know. Maybe it is, maybe it isn't. But for sure, the infarct getting bigger and the heart dilating, those are both bad things.
And what kind of time frame are these pathways active post-infarct? Like a week, or just a couple?
Yeah, I think
days,
those studies are hard to do in terms of the exact timing of it. But I think they're certainly active for several days, and we saw that P38 was still active a week later. But whether it goes, and we didn't do a careful study for months to say, does it turn off at 2 weeks or 4 weeks? So I don't know how long it lasts, but it certainly lasts for several days to a week, at least P38. And I'm not sure about, I don't know how long, we showed that TRP3 was induced at 1 day, but I don't know how long that lasts, so I'm not sure yet.
Do you know anything about their role in myocarditis, for example, like infectious? Anything where there's a lot of cell apoptosis in the heart?
Myocarditis in general is that's a situation like what Catherine has brought up before, where inflammatory cells are the whole story. With myocarditis, really inflammation is the key, and the inflammatory cells create a variety of issues with myocarditis, and to me that's a different category of disease than what I'm talking about today.
Just a quick question.
Yes.
Are these signal pathways related to the modeling confirmed by human heart study?
We know that several of these signaling pathways are activated. Certainly in hypertrophy, we know a lot of them are activated. In heart failure, a lot of these things change as well. In the setting, the acute MI setting, I'm not so sure that we know. It's technically and ethically challenging to get heart samples in that period. I suppose if somebody went to the operating room, we could try to get, but you almost never get left ventricular samples. We can get like atrial appendage samples, but it would be tricky. I suppose if somebody died and you get an autopsy specimen quickly enough, you could do it that way. But it is. I agree with you. That's an important consideration, and we do need to. There are, as you know, there are ways now of imaging these molecules in vivo, and so there are ways of looking at activated ERKs or activated AKT in vivo. And in fact, the Groppler group is working on generating these in vivo scans by PET to look at activation of these molecules. Although I don't know how good the resolution will be, but there are people are trying to work on doing in vivo imaging in humans of the activation of these proteins.
So is it possible that some signal pathway during the remodeling could be species? You know?
Right. No, it could be. It could be. Yeah. Okay. Thank you.